Hello, everyone. How are you? Thank you for joining. Welcome again to everyone joining from Australia and around the world. My name is Shalin Naik, and I'm a group leader at WeHi. So I'm joined today with Helen McGuire from the University of Sydney and the Ramaciotti Facility for Human Systems Biology. Hi, Helen.
We get there eventually. Hi, Shalin. How are you?
Good. How are you?
Good.
Right, so I want to begin by acknowledging the traditional custodians of the land on which I meet with you from today, and this is the lands of the Wurundjeri people, and pay my respects to their elders past and present. And I extend that respect to the traditional owners on the lands on which you all meet from, and pay my respects to any Aboriginal and Torres Strait Islander peoples here today, especially because this week is NAIDOC week. So I hope everyone is getting involved. So I'll now just share my screen for the sponsor slides. So just to once again thank all of the sponsors who have contributed to this series so far. This is actually episode 9. And this online series has been really successful this year. We've got a typical reach around 200 of you logging in per episode. So thanks for your continued support.
Sorry there, Shalin. I'm just going to get you to swap your screens. We were seeing your notes.
Well,
try again.
There we go. Let's try that.
Fabulous.
Well, now you know what I was going to say, don't you? So today's episode is kindly sponsored by BioLegend with Australian Biosearch and Scientific. So thank you very much. And we're going to hear from 3 wonderful speakers today: Ellen Rothenberg, Ashraful Haque, and Joel Rimes. We'll hear from our 3 speakers with time for 1 or 2 questions immediately after their talk, and followed by a longer 25-minute Q&A at the end. So please fire away with questions in the Q&A panel you'll see in your screen, in the top right of your screen. And you can ask a question with your name or anonymously. And we have enabled the upvoting function for questions of others' questions that you might like. Also, please engage on Twitter with the hashtags OzSingleCell and SingleImmuneCells. Your random thoughts, ideas, a picture of your cat, whatever you like, always welcome. I'm going to now hand over to my colleague, Dr. Helen McGuire, to introduce our speakers today.
Great, thank you. So Shalin, that's a wonderful introduction, and I think this is going to be a great discussion today. So I think what we really wanted to do was just get started, really. So we've got a great speaker lineup, and it brings me really a joy to welcome a hero in our field, Professor Ellen Rothenberg, to start us off on this conversation, as it would be, of single-cell technologies and immune research. So Ellen has been in research for a number of years now. She's a professor of biology at Caltech, the Alfred Billings Ruddock Professor of Biology at Caltech. So having graduated at Harvard University, receiving her PhD at Massachusetts Institute of Technology. She's undergone some postdoctoral fellowship training at Memorial Sloan Kettering Cancer Center, and taking on a faculty position at Salk Institute. From there, she's gone in leaps and bounds and has been awarded a number of wonderful accolades along the way. Is a Feynman Prize for Excellence in Teaching, is a fellow of the American Association for Advancement in Science, and a fellow of the American Academy of Arts and Sciences. So a real pleasure to welcome her. She studies gene regulation and development of T lymphocytes, gene network controlling hematopoietic cell fates, and mechanisms underlying the dynamics of single-cell development decisions. So with that, Ellen, I'll welcome you and ask you to share your screen and tell us a little bit about what you're working on today.
Thank you so much. First of all, I just want to tell you how delighted I am to have been invited to participate in this, and I hope it will be enjoyable for you. It's a little bit different from a lot of the immunology that you hear about, and even a lot of the applications of single cell technology, which are more medically oriented, we're really interested in the basic science of cell type development, and specifically how you can make a committed, incredibly durably committed T cell out of a cell that starts out as a hematopoietic multipotent progenitor. And so I just wanted to share some work with you, mostly from one graduate student in my lab, Wen Zhou, who has been pioneering our work in this area. Now, the background of the system that we work on is in the thymus. I'd like to contextualize the stages we're going to be talking about. This is all before T cell receptor gene rearrangement even begins. So these cells are not T cells yet. They're basically right on this boundary from the multipotent progenitor that goes into the thymus and the process of commitment through which they give up all of their other developmental alternatives other than becoming a T cell. This process occurs under the influence of Notch signaling. That's the absolutely overwhelmingly important signal from the thymic microenvironment. But the important thing is that it's a long process, and it accompanies a great deal of cell proliferation, basically 10 days of cell cycling on the order of, at least one, usually 2 cell cycles a day. So the question is, why does this take so long, and what is going on in this process? And at the end of the commitment process, although there's still a lot of T cell development left to go, basically the essential features that give cells a T cell identity are already established at this point. Things like CD3 expression, kinases that are going to be used in TCR signaling forever after, and they're never turned off again. So this is quite important. Now, over the years, my lab has been focusing on these early stages, and this is when commitment occurs right here. But what you can see is that this is quite a complicated process. Down at the bottom, you can see the expression dynamics of a whole bunch of different transcription factors that we've actually followed through this process. And you can see that there are both activations and repressions of many transcription factors right in this commitment window. You can also see something else that we're quite interested in, which is changes in sort of the pattern of epigenetic chromatin accessibility. I won't have a chance to talk about that today, but if any of you are interested, we could talk about it afterwards. On the other hand, the whole question of what this mechanism is, which my lab is studying, where does this transcription factor bind? Does the site open or not? Does the target gene go on or go off at that point? It really all depends on having the story right about what the starting point is. And that really involves having an unambiguous definition of these earliest events, which are the most poorly understood of all of T cell development. And even exactly what precursors they come from before the cells get to the thymus. And so to deal with this dark phase of T cell development, Wen Zhou in my lab, a wonderful graduate student, really embarked on doing this properly at the single cell level so that we could attempt to align specific cell transcriptome states with developmental potential states and with stages along a developmental progression. And essentially, it was already known from our bulk population studies that the earliest population in the thymus, the ETPs for early T cell precursors, had bulk gene expression that really resembles multipotent progenitors in bone marrow, really LMPP-like gene expression, not even CLP-like, it's really LMPP-like. But is this because these cells are really like MPPs, or is it because they're intermixed with irrelevant cells from other lineages? And also this comes along with the question, if they are expressing LMPP or MPP genes, that means they're expressing still a lot of genes which are implicated in hematopoietic stem cell homeostasis. And do those genes have a legitimate role in T cell development, or are they just passengers? Are they just because we couldn't purify out that initial state well enough? And does this give us any clues as to the kinetics of the process? Could it be that they're actually competing gene network circuits, driving the cells forward and others trying to push them back? And so Wen decided to really select cells across the continuum of the earliest developmental stages in T cell development using well-established markers, KIT and CD25, to isolate the 0.01% of thymocytes which are in these stages. And as an outgroup, to give polarity to the gene clustering, she sorted a very small percentage of very purified DN3s, which are the cells that are in the process of doing their first T cell receptor gene rearrangements. We then characterize these cells by not just one, but 3 different single cell transcriptome methods. Of course, we used 10x droplet-based single cell RNA sequencing, but we also complemented this with a deep sequencing from Fluidigm into deep sequencing. And we also used, very importantly, a single molecule fluorescence in situ hybridization multiplex process called seq-fish. This was the original version of seq-fish. And if you are interested in the details of what I'm going to talk about in this first part of the talk, they're all from this paper that was published just about a year ago. Our goals in doing this was obviously to dissect subpopulations, and we wanted to order them, but we also really wanted to determine if any of them are branching to or from non-T cell fates, so that we could actually avoid confounding the T cell progenitor program with other genes which are associated with cells doing something else. We wanted to specifically zoom in on the regulatory genes that are active and relevant in the T cell precursors at each stage. And this is why we needed to do some other modalities besides droplet-based transcriptomics, because we needed the depth so that we could get to low copy number transcripts, so that we could get accurate readings on transcription factor coding genes in individual cells. And we wanted to make sure that the pseudo-time trajectory we would develop would be correct, and we wanted to validate this with actual developmental time courses, as well as computational methods, and hopefully be able to hypothesize control functions for some of the developmentally controlled regulators, which we could then test. So I'll just sort of give you a quick spin through this data over the next few slides. Now, the first thing is that if you plot the data from the 10x data sets in a force-directed graph, this is Spring from Alon Klein's lab, and you look at the populations of cells, the way they self-organize, this is totally unsupervised. What's very nice about this process is that although the cells are proliferating throughout the whole process, the cell cycle axis is basically orthogonal to the developmental axis as it's plotted in these first 2 dimensions. So essentially, the cell cycle is going from the G1 cells at the upper left to the G2 plus M cells at the lower right, whereas the early stage expressed transcription factors are all the way across the upper right, going down to the latest activated transcription factors at the lower left. And so essentially, we can look at these just as a cross and see these different properties. You can also easily see some outlier cells emerging here. These cells, when we purify them, prove not to have anything to do with the T cell fate, so we can exclude them from later analyses. Now, the question of transcription factor genes, of course, is at the core of our program. And because transcription factor genes are expressed usually at less than 50 copies per cell, often less than 20 copies per cell, 10x is very poor at actually detecting them accurately. And we found that by using this multiplex single molecule FISH, where we could look at something on the order of 65 regulatory genes in single cells in these analyses, we found that we had about 10-fold higher sensitivity for these low copy number transcripts than the 10x analysis. And this allowed us to correct the dropouts so that for genes like Notch1, Runx1, TCF7, that we know are not only expressed, but absolutely crucial for the viability of every cell in these developmental states, we could see them 100% positive with the seq-fish, whereas there were between, 40 and 60% false negatives for these same genes in the same population with the 10x, whereas we could still accurately see the bimodality of other transcription factors that were being turned on from an off state in that population. And some of these data allowed us just to get some good confirmatory data. So for example, GATA3 and TCF7 do start to be expressed from the earliest stage, these ETPs on the left, whereas BCL11B is not expressed at all in the ETPs. This is the background level, and it's only turned on at DN2 quite robustly. You can see that the cells expressing BCL11B are all cells that have already turned on TCF7 between 25 and 50 copies per cell or more. GATA3 is at a very low level at ETP, but always in cells that are all expressing TCF7, and they get upregulated in DN2. This is totally consistent with what's known about these genes from bulk populations, but now we're seeing it in all of the cells. And we can also see things like, is the T cell program shown by the GATA3 and TCF upregulation, is this actually happening in the same cells that are expressing progenitor-associated genes or not? We can use one of the earliest progenitor-associated genes to go off, LMO2, and this gene is actually still being expressed in the majority of the cells that first turn on TCF7. The dark blue ones are the ones that are in the ETP compartment turning on TCF7. At least half of those are still expressing LMO2 at that time. Even some that turn into DN2s are still expressing, whereas BCL11B is only turned on in cells, except for this one, that have already turned off LMO2. And you can see this very, very clearly. So this is a tremendous help then to clarify gene regulatory network circuitry, which otherwise might be sort of stuck in a gray zone. Now, this allows us then to go with some confidence to build a high quality pseudotime model. This particular model is a supervised pseudotime model, and it's based on pruning out those outlier cells that we already proved are not in the T cell lineage. But what you can see is that it has some features that we expected. That is, the progenitor-associated genes are clustered here, and they go off. Red color is going to blue as the cells go forward in pseudotime. The T cell specific genes really come on at the end, and they go from blue to red, as they should. But what was interesting to us was that that's not all that's happening. And what you can see is, I think there's at least 2 different waves of other genes which actually get transiently upregulated and then downregulated, then a little bit later upregulated and then downregulated, which seems to hint at a more complex process with maybe more regulatory staging step by step by step than the simple get rid of your progenitor identity, turn on the T cell identity model that we started with. Now, one thing that made this very interesting is that the genes in this first transient wave are actually very enriched for myeloid associated genes. PU1 encoded by SPE1, TYROBP, MPO even, myeloperoxidase, and the genes that are upregulated in the second wave tend to be enriched also for some innate lymphoid cell genes. So one question is, this is a pseudotime is a computationally derived model in which you're trying to force gene expression transitions into a linear pathway. And one question is this correct or not? And so is this accurate? And one way to look at this is to use RNA velocity and ask whether it basically supports the model. That is, if you look at the flux of cells based on the ratio of intronic containing RNA transcripts that haven't been spliced yet versus the fully spliced mature transcripts, does it suggest that the cells are newly turning on the genes which actually belong to the correct path predicted by the pseudotime trajectory? And the short answer, I'm not going to show you the data, is yes. We also correlated this with the known acute effects of removing some of the known regulators that we've already played with in perturbation studies around the commitment checkpoint. And to the extent that we were playing with the right regulators, we indeed could see some enrichment for the positive and negative regulatory targets of those regulators around the transitions predicted by the pseudotime to correspond to those regulators going on or off. But what I wanted to show you is the thing which I think is the most important, which is that we actually decided to use the pseudotime to identify population subsetting cell surface markers that would allow us to subdivide these populations into smaller bins, and then ask critically, if we sort those live cells out and race them against each other, do they actually behave as though they are in a linear order of T cell developmental progression, or are some of them really branches? And so just to show you what I mean, if those myeloid genes that are turned on in that first wave are really T cell progenitors, then the suggestion is that they would actually be closer to T and further from multipotency than the cells before them in the pseudotime. However, if it was just a computational artifact from the computer clustering method, and if these cells were actually a separate branch to myeloid, then they would tend to have less T cell potential than the others. So by actually selecting these cells out and then challenging them with in vitro cultures under T cell or non-T cell developmental conditions, we can then ask after a fixed amount of time whether these cells have the right developmental properties as predicted by the pseudotime, or if it's something else. And this is just an example of what this looks like on a bulk population level. If we use the pseudotime genes to split the ETP population, this is all ETPs before the cells even turn on the first definitive T lineage marker for DN2. These are all ETPs, but they're subdivided now according to these stages. And we can take bulk populations and race them against each other. And this is after 4 days of culture. And the milestones we're asking them to cross are first this one and then this one. And you can see that the cells that are the supposedly earliest, almost none of them get to the second milestone, but almost all of them get past the first one. That's good. That says they're in the T cell pathway. But whereas the most advanced, almost 93% of them have gotten past the second milestone. The nice thing is that the ones in between, including the ones that we think are upregulating the myeloid genes, these populations are actually behaving like intermediates. They actually are faster at getting into the T cell pathway than the earliest population. So that looks as though it supports. We can do this experiment over again at the single cell level by not only taking those sorted subsets of subsets, but we can actually seed clonal cultures with 50 to 60 of each, 50 or 60 individual clones from each of those subsets, and then look at the statistics of those clones, whether you're basically having all of the development coming from a few outliers, or whether it's really coming from representatives of those clones. And what you can see is, again, step 1, step 2, step 3 of development. All of the clones, no matter how immature, mostly get every single one of their clones past the first milestone, but really while the most mature get almost everybody past the second milestone, the ones in between really have intermediate levels of success in getting to the second milestone. And for the third milestone, which I didn't show you before, the most mature have a lot of clones that individually got to the first milestone, whereas the earlier pseudotime subsets did not get there at all. And if we go back and take those same sorted subsets of subsets and then redo the single cell RNA-seq on them, we can actually validate that this does go along with transcriptome population behavior that looks as though it is actually in the right order in the pseudotime. So we went to the trouble of this validation because we really wanted to know if these transcription factor dynamics are right. And essentially what you end up is this picture that you actually have several overlays of different transcription factor dynamics going down and coming up. And this basically agrees with what we had seen at the bulk level, but now we know it's actually true. So we can now use this confirmed knowledge and the single cell method to try to look at which of these factors are really important to control the actual dynamics of the differentiation process, not only at the known commitment checkpoint itself, but actually in these earlier stages. Does anything happen earlier that depends on these factors?
Hi, Ellen. Just a little bit of a time check there. If we can start to think about 2 minutes or so. Great.
That sounds good to me. So I'm just going to tell you, in the system that we have for in vitro differentiation, we can actually do either deletions or add backs of these transcription factors. And we decided to apply this to some of the transcription factors that we thought were important seemed to be important in this study. We used a double guide RNA knockout modality with barcodes that would allow us to tell which of the transcription factors had been targeted by the guide RNAs infected with a particular construct. And then we could pool the cells that were infected by a whole library of different guide RNA coding cells in the same single cell analysis. And by taking the cells for this analysis from Cas9 transgenic mice, we didn't need to add anything except the guide RNAs in order to get deletion. And the results actually show that we could use then perturb-seq with which in a site-seq type of pooling, we actually had batch control. Everything was in the same batch with internal control vectors. And you could see I won't have a chance to run the movie for you, but if this is the distribution of stages in the control across pseudotime based on the single cell analysis, we could see that the different perturbations actually, some of them did almost nothing to the cells, but some of them really affected their average transcriptome and others affected in the opposite direction. And I think that I can just show this to you here, whereas if this is the control, and this is a 2D plot of this in UMAP space, the controls are all here, where the early end is here, the late end is here. If you knock out the gene that is important for T cell development, the cells all get stuck in the early end. They don't get past the early end. If you knock out PU1, however, the cells go faster, and no one is left in the early end, not even as many as are there in the controls. You have other genes which do other versions of the same thing, but not quite the same way. GATA3, also a little bit biased towards the early end, this HOXA9, but the champion was ERG, a very interesting transcription factor that's been studied in Australia in Benjamin Kyle's group, a critical stem and oncogene. And it turned out that ERG has roles not only in keeping the cells in this progenitor state, but also in making sure that they stay later in the correct control pathway. Without ERG, not only do they go a little bit out of this progenitor state, but they fly into this completely aberrant state and completely cross, suggesting that this factor both has a role in early progenitor status, but also in guiding, collimating the correct pathway for T cell development. So the conclusions are just that the T cell signature transcription factors are activated in cells that are still expressing this multipotency program. And I'm showing you at the end, that's actually relevant for correct development of the cells, not only in terms of timing, but also in terms of direction. We validated the trajectory, and the perturbation analysis shows that there are actually different kinetic pushes on the process coming from the progenitor associated factors on the one hand versus the T cell factors on the other. And so again, this work that I summarized was mostly the work of one graduate student, Wen Zhou, with help from Mary Yui, who is a senior research professor in our group. We had many marvelous collaborators who I've listed here, and our support for most of Wen's work was from this grant from NIH. And I apologize if I went over time. I hope there's time for a bit of questioning, but if we don't, then there's time later on, hopefully in the discussion.
Thanks, Ellen. That was powerhouse and right up my alley. There were so many beautiful controls and validations. I love it. I love it. We can take one question, I think, from the audience, and then we've got a couple of ideas that we might actually bring to the panel discussion at the end. And so we've got a question here from Charles Bell, who was asking whether you had a hypothesis. What do you hypothesize as the function of the upregulation of these myeloid transcription factors in the early phase of commitment? And do you think that these transcription factors have pleiotropic function, or is it that they could potentially have something to do with your previous observations about transcription factor theft?
Haha. Okay. So you read our papers. This is very kind of you. Thank you. PU1 is one of the factors, even though it's expressed right at the beginning, it actually does undergo a slight increase in expression. And we really we know that the cells are getting activated by the Notch signaling, and we know that PU1 itself is a little bit activated by Notch signaling, which is a surprise to many people for the gene network. But we really don't understand why this should be unless they are being used pleiotropically to try to accelerate the entry into the cell cycling. PU1 knockouts certainly slow down proliferation, and at least one other transcription factor that's upregulated in this group that another graduate student in the lab is studying seems to have a role in accelerating proliferation at this stage. So we think that the myeloid side effects are really just side effects of the benefits of these transcription factors to support proliferative expansion, and possibly also to prevent the cells from undergoing commitment too quickly, because the commitment is linked with an anti-proliferative gene network circuit. And so it may be that the cells would have to pay a price if they went through commitment too fast.
Yeah,
thanks so much for the
fail
great
safe.
Yeah,
Excellent. What a really interesting idea. Really great. So thank you, Ellen. We'll move along to our second speaker, and we'll look forward to discussing more with you as we continue later on. So Dr. Ashraful Haque Ash has received his PhD in 2002. He started his research career at the Imperial College in London. And he started out looking at salmonella bacteria, moving on to studying how immune cells protect against in the situation of lethal sepsis. It's in 2006 that he became known to us in the Australian community of immunology, where he moved to Queensland. He was at QIMR in Brisbane and received NHMRC funding in 2010, starting his own group in 2012, and has since been recruited to the Doherty Institute in Melbourne at the start of this year. So congratulations, Ash. So his group is focused on T cells. So we're keeping on the T cell theme, CD4 cells, and how they are influenced, their influence is involved in cancer and infectious disease. And I believe he just likes to get as much detail into his analysis and study as possible. So with that, we'll invite Ash to share your screen and tell us a little bit about your story. Thanks.
Thanks, Helen. Thanks very much for the invitation to speak today. I'm going to put my slideshow on, and going after Ellen, you can see some similar themes, but we're going to be pretty light on data, so I apologize for that, but I would encourage any questions to grill me on the background and the detail. So my team and I are really interested in cellular change, but not in the thymus, more so in secondary lymphoid organs in the spleen or lymph nodes, looking at how T cells acquire skills. And in thinking about that this morning, I realized that I was, pretty much like these gray cells 2.5 years ago when it came to a particular functional capacity, that of swimming. So I could not swim really 2.5 years ago, but something gave me a stimulus around that time, and I began to try and learn this skill. And maybe about just over a year ago, I could swim. I could swim approximately 1 kilometer without drowning, which was a great achievement in my book. And then we moved to Melbourne, and then the pandemic hit, and I didn't swim for maybe 10 months, I think it was. And the question that I was considering about a week ago is, had I retained any of that functional capacity? And so this morning I jumped into the pool, and lo and behold, I completed 1 kilometer of swimming. So just like these T cells, I'm interested in how I can acquire skills and how I can retain them. And of course, that's what we're looking at for T cells. Our interest in single cell genomics started maybe 7 years ago. And we were interested in CD4 T cells in experimental models of malaria. And we were able to capture transcriptomes of activated CD4 T cells. And over time, using different modeling processes that I'm not really going to go into any detail, but it really involved a two-step process of dimensionality reduction and which of course involved a machine learning approach, and then another process called overlapping mixtures of Gaussian processes to find our way, if you like, through the data to generate a probabilistic model of that data. But our central interest was, how do T cells preserve memories? How do they retain their ability to function after their initial learning of a skill? And so more recently, we wanted to essentially repeat that same process, but follow T cells not only from naivety here in grey and through the first week of infection, but really what happened beyond that for the rest of the month. And using exactly many of the same techniques and computational approaches that the previous speaker used, we were able to generate a model of how cells might choose between two fates, either swimming or scuba diving, and whether or not they retained any of those skills at the end of the month. But a model is of little use unless one can start to delve into it. And just like we might be interested in gene A or gene B, we wanted to look more at the dynamics of every gene. And we used a method from Valentin Svensson, spatial DE, to really categorize every cell or every gene rather, according to the dynamics of their expression over that month-long period, which is what I'm depicting here. And of most interest to us was this red line. Genes that were upregulated but then preserved to a certain degree at the end of the month. Perhaps these might be of interest in terms of characterizing memory preservation in T cells. And I think it was of interest that only approximately 3% of the genome was really characterized with this dynamic. And having done that, of course, we wanted to look in more detail, and that's really what our single cell based models allow us to do. And so I'm not going to go into any of this detail of this network, but it allows you to look at correlations between genes in individual cells. And by doing that, for example, one can make connections between genes that are well characterized in the literature and well known, and newer genes that perhaps have been poorly characterized or not characterized at all. So it's very much a hypothesis generating exercise and to provide a resource to the community. And so that's what we've done. You can visit our URL, our app generated by our team, and you can look at the data and you can choose the model that you're interested in. You can, let's try that again. You can choose the model, you can type in a gene of interest and look at the expression dynamics in our model. Because it's a SmartSeq2 data set like Ellen introduced, it's high resolution. You can look at co-expression of two genes. And that helps you really sort of solidify your own hypotheses about your gene, your favorite gene. But you can also tap into the second independent data set on our tab to look very specifically at the end of the month, to help you really look at minor differences, subtle differences between a range of different inferred cellular states. So that was a major end goal for us, and we would encourage you to use it if that fits with your interests. But of course, we are increasingly wanting to supplement our transcriptomic data with other types of information. I'm showing here, superimposed over our model, green lines that connect T cells that have the same, that are siblings, that have come from a single a single naive T cell, clonally expanded, and we can detect the siblings. And that's obviously of great interest for T cell biologists to understand the relationship between TCR and fate choices made by those T cells. Now that's based from a high resolution SMART-Seq2 data set, but what I think is really exciting nowadays is that so many of us at least have access to a droplet-based system, often within our own institutes, which is this sort of this toaster up the top left, the Chromium controller. Now, I'm not into product placement, and I didn't mean to put a Becton Dickinson machine and an Illuma machine. You can use your own generic home brand cell sorter or sequencer if you so wish, as we do. But the point is that you can generate your own data. And of course, the expense is an issue, but you can generate it. But now we're looking to supplement transcriptomics with other types of information. And one of those types of information that many of us might be interested in is not just the protein encoding islands within the genome, but all the other massive expanse of important DNA, enhancer regions, transcriptional start sites, and the like, which we can perhaps get some information on at single cell level using chromatin accessibility information. And we certainly wanted to understand how chromatin changed over time in our T cells in a model of malaria. And we didn't use a droplet-based system. At the time, we used a plate-based system, which allowed us to look at a certain number of cells, but not many, many thousands. But it really painted a very similar picture to the transcriptomic data, which was that compared to naive cells, we see a massive increase in heterogeneity in a variety of effectors, and that transition to memory constitutes a partial coalescence and return to naivety, but only a partial return. So we really hope that by combining chromatin accessibility and transcriptomic data in the next months and years, we may get a fuller picture of cellular change, not only for the T cell system, but any cell of choice. But so much of this is looking to, if you're thinking of yourself as a T cell as introspective, it's looking in great detail at that cell, and to anthropomorphize even more. It's not necessarily what's going on inside you that dictates your fate, but more what interactions you have with those around you. And if one were to read a biography, and this is a particularly good one that I recommend to you, one doesn't necessarily always dwell on the internal. One is often interested in the external influences that push you towards one fate or another. And so in this microscopy image of the spleen, you can look at these thousands of GFP cells, these thousands of David Attenboroughs wandering around the environment of the spleen, receiving interactions from like-minded cells in red, or distant relations like B cells in blue. And so they will dictate, as we know in immunology through a wide variety of different approaches, static microscopy, intravital microscopy, that these cells are important. But we really wanted to move our transcriptomic data with spatial resolution, and to do so in an unbiased way. And so we really have been working with Fei Chen at the Broad Institute, who has a wonderful technique called Slide-Seq, which he published in Science a couple of years ago. And in this UMAP, I'm showing you individual beads. Each dot is a bead, not a cell. And that bead is immobilized on a flat array, a puck, as they call them. And you place your tissue on top of that puck, and you harvest very locally the mRNA from cells overlying those beads. And what you're seeing here is the beads colored according to the transcriptomes they've captured. And we know, for example, that these light blue cells are B cells. And we're not looking at the spleen here, we're looking at the gut, where we knew there were some immune tissues, like Peyer's patches. But if you reposition those beads from the UMAP to where they actually are located on this circular puck, you can see some beautiful detailed structure, which allows you potentially to ask questions around the interactions between cells, and by diving into the transcriptomic data, perhaps finding some novel data. So we've since started to try and look at the spleen. It was important to use a high resolution method like SlideSeq because the spleen is so dense with cells, we need that detail. And again, here we're looking at beads colored according to the transcriptomes they've captured within the spleen. And we're hoping that we can start to find some interesting novel interactions between any cell you might imagine within a secondary lymphoid organ. But it's not proving as easy as that. And one problem that I'll share with you now in the last minute or so is one can imagine one bead on that array, and cells are not placed nice and neatly over that bead. What's more likely is that two cells will partially overlap with that bead. And so that bead will capture mRNA from two different cells or two cells. Now, if those cells are very different, that bead, the data from that bead, can easily be flagged as a doublet. And if you're into flow cytometry, you might throw away that data. But we might have an extra trick. And of course, we know that there are other beads, neighboring beads on that array. And we might be able to essentially perform a subtraction, a removal, and some data manipulation, if you like, to examine and subtract the data, deconvolute it into the individual cells. So that's one of the major challenges that we're working through. And of course, there are algorithms developed to work on this. And the other major challenge would be that compared with high resolution single cell RNA sequencing or droplet based, which gives you thousands of genes to look at, currently we're still working in the many hundreds of genes per bead. And that may or may not give us the resolution that we require to discover new biology. So with that, I'm going to stop. I hope you can forgive me for being light on data. I'll be happy to take any questions you might have.
Thanks, Ash. That was fantastic. And I think I've got my quote that I want to put on my t-shirt for today, which is thousands of David Attenboroughs wandering around the spleen. That's my, I was inspired by that. Great, great analogy. So we do have one question from the audience that we can press on with straight for you. And that is, you discussed the siblings by TCR arrangements. So the plots where you showed the sisters, I think you referred them as brothers, were connected. Some were both in effector cell clusters, whereas some were across concordant in different fates. Can you elaborate on these findings from your analysis?
Some of them were in, so the way that experiment was done was harvesting experimental mice at various time points. And therefore, we can only really examine siblings within a range of different states that were present within that mouse. So it's not easy to see, or you can't see any relationships between very late in pseudotime versus very early in pseudotime, because unfortunately the mice were completely separate, and so you don't get any sharing of those TCRs across. Now, I'm not sure if that was a direct answer, but I wanted to explain how we got to the data. Yeah. Okay,
cool. All right, and then I think the work that you were doing with the SlideSeq was just really phenomenal, and we're all excited to see where that goes. And Shalin actually had an idea or an concept whether you could study using the CD45.1s and 0.2 chimeras for spatial arrangements, and could you do something like sort out by hashtagging to separate the doublets, so get around that or understand that idea a little bit?
Can you hashtag to separate doublets, I guess is the question. Possibly, or if the chimera was 50-50, then one would assume that you could game it so that there was a random allocation of 45-42 across all of the splenic cells. And so you might have, for any cell, maybe you would assume that 50% of the cells surrounding it would be of the alternative congenic. So that would perhaps reduce your problem. But I think, yeah, it could help, but then of course a chimera has its own issues as well, right? So,
yeah. And I guess you've always got that issue. You don't really want to throw away doublets necessarily. You just want to understand them, I guess. So, yeah. Okay, well, we've got a couple more ideas popping through, and we might bring that to the Q&A question. Thanks, Ash, for a great presentation and a great segue into Joel Rimes' talk. So, Joel, today I will invite to present. He's a final year PhD student with Associate Professor Edwin Hawkins at the WEHI. So his research applies various microscope techniques, and that allows him to look at long-term tracking of immune cells and autoimmune disease development in vivo. So I think he's going to tell us possibly about a couple of projects, but he's looking into investigating the role of plasma cells in lupus, as well as 2-photon imaging and single-cell RNA-seq data to better understand bone marrow biology. So Joel, welcome.
Thank you, Helen, for the intro. Let me just share my screen here. Hopefully that is showing the screen that I'm looking for. So yeah, I'm a PhD student in Edwin Hawkins' lab at the Walter Eliza Hall Institute. And as Helen said, my research really focuses on the use of in vivo multiphoton imaging to better understand the dynamics of plasma cell function in the bone marrow. So a lot of my studies are done in the context of systemic lupus erythematosus. So I'll just very quickly introduce the disease setting on which I'll be showing you. It's a complex polygenic autoimmune disease. Because it's polygenic, no two patients are the same. We get great variation between patients and both within patients, including periods of remission and flare-up. The causes are thought to be a combination of environmental and genetic factors, and this results in an array of symptoms, including the characteristic butterfly rash shown on the slide, but also more serious symptoms, including painful and swollen joints, fatigue, and glomerulonephritis, which is antibody complex deposition in the kidneys and can lead to really serious kidney disease and kidney failure, and can also force patients onto dialysis. It affects about 1 in 1,000 Australians, and this incidence rate is 4 times higher in the Indigenous population. And currently there is no cure, and the treatment options are very non-specific and very limited. Usually the use of glucocorticosteroids or large-scale immunosuppressive agents, which can lead to very severe side effects. So SLE is driven by high levels of antibody production that targets healthy tissue. And of course, this antibody is produced by plasma cells. So we have the standard B cell response in which naive B cells encounter self-antigen. This can lead to plasmablasts, which create antibody, or germinal center B cell formation, which can lead to plasma cells and memory B cells. So my project focuses on understanding how these autoimmune B cells behave in vivo, particularly using multiphoton imaging, and what they interact with and how they can be targeted by therapies. And I have a particular focus on the microenvironment of these cells in the bone marrow. So a few points on my research. I'm very interested in how these cells behave, both in the bone marrow, but also in peripheral tissue, and using the imaging techniques to better understand that. And also, as I said earlier, the plasma cell niche, which is quite a controversial and disputed topic. There are many different models which suggest many different components of the plasma cell niche, including cells like megakaryocytes, eosinophils, T cells, DCs, stuff like that. So I'm hoping that I can use multiphoton imaging to better answer the questions around these diseases and these niches, to better understand, for example, the existence of the niche, the composition, whether they're dormant or dynamic in nature. So in order to investigate that, I do most of my research using a mouse model, the MRL confetti AID Cree mouse model. Essentially, in summary, what we have here, the MRL LPR component is the disease component, which is very similar to the pathogenesis we see in humans with SLE. And the AID-free confetti leads to these mice developing these cells which can carry one of four fluorescent proteins, being GFP, RFP, or CFP. And what this leads to is post the expression of AID in the germinal center, we get post-germinal center B cells that are labeled with one of these colors. And these colors are conserved as these cells divide into their daughter cells. So just to show the characterization of the model, on the left here, I've just got a breakdown of the confetti-positive population in various compartments, just demonstrating the correlation between CD138 expression and size, shown by the forward scatter, which will be important when I show you the imaging data later. You'll see how size plays an important role in these cells, and also how they have a higher degree of fluorescence, shown on the bottom left here. Again, that'll come up later when I show some images. And on the right, just some ANA data demonstrating that there's a clear autoimmune phenotype in the aged MRLs as compared to various controls, including aged matched healthy mice as well as young. So here's how the confetti population skew manifests in these mice. Just moving down the plot here, we have different mice showing their bone marrow and spleen, and you can see that we can get very different proportions both between and within mice, which suggests that the makeup of these proportions are due to the expansion of particular pathogenic clones that grow out and skew the population throughout disease development. And on the right here, I've got some examples that show us that this variation can once again be seen within mice in different bone marrow compartments. So again, moving down the page, I have 3 different mice shown here, and moving across just how different long bones look. And you can see these populations, there's always a general profile between mice, but you can see there's a real tendency for these to skew quite extremely, as shown in the right femur of that second mouse there, which is likely due to the expansion of a really dense cluster of RFP positive pathogenic cells. And I'll show some images of examples of that in a moment. So like I said, it's all about the multiphoton imaging to get that live look at the data. So just to briefly introduce the technique, which was developed by the head of our lab, Edwin Hawkins, using 2 multiphoton microscopy to allow us to look in the bone marrow space in a live dynamic type of way. And through some surgical intervention on specialized apparatus that we have on hand, we're able to create an imaging window into the bone marrow space of the skull. And what this allows us to do is image small stacks as represented here on the left, which we can stitch together to give us what we call tile scan data, or also continually image small areas to give us what we call time course data. And the beauty of this technique is the capacity to image over time, both in a single session for a number of hours, but also over multiple sessions or on various days up to a few weeks, allowing us to capture the same areas and monitor them as they change through periods of disease progression, therapeutic intervention, or whatever else we're introducing into our model. So let me show you a brief summary of how that data looks. Let's just start off with some controls here. This, for example, is just a young MRL confetti mouse before it's had the opportunity to develop disease. So these mice demonstrate no autoimmune symptoms, and you can see here, it's largely just bone signal. We don't really have any confetti positive cells. Another control are our aged healthy mice, so our AID-free confettis without the MRL component. These mice are also relatively healthy with no autoimmune symptoms. And here we can see the presence of a few confetti positive cells sparsely dispersed amongst the bone marrow, but again, we don't see any expansion or clustering or real disease in these mice. Next, I'll show you how these healthy mice look when they respond to immunization and boost using MPKOH primary immunization. And I don't want to go into too much detail in the interest of time today. So essentially, I'll just give a brief summary, and what we see here is a slightly larger degree of confetti positive infiltration in these mouse. And we begin to see examples of these small single color clusters form. You can see, for example, a small green cluster up there on the top left. But this is something that we see far more pronounced in our SLE model. So here, these mice are very sick. They have very enlarged lymph nodes and other autoimmune symptoms, and we see much denser confetti positive infiltration. So we always, or very often, we have these single color clusters in the bone marrow. And these are much larger and more frequent than what we see in our immunization model. And again, they are always single color. And this also looks a lot like the biology that we see in the immunization, but far more extreme, which when we step up again to a multiple myeloma model, you can see here, we have a very similar phenotype with huge degrees of infiltration. Again, these outgrowth of single color dominated clones with very similar characteristics for what we see in the SLE. And I won't go into detail on this today. I'll be focusing on the SLE. And all this multiple myeloma work is done by a fellow PhD student in our lab, Joy Lu. So here's just some zooms on some areas of interest. Those mice, for example, you can see in the control mice, we have very few. And then as we step up in our models, we start to see larger single color dominated clusters and clones expanding. So from here, I'm just going to focus on this SLE. So just to give you an example of how some other mice look and how this data comes out, give you a look at what these clusters look like in three-dimensional space. And another different mouse shown here. And you can see from these examples that we get a great degree of variation in the distribution of our confetti-positive populations between mice. However, there is always these conserved characteristics. Conserved characteristics such as the single color nature of these clusters across all of our examples. So the beauty of this technique, as I said earlier, is the capacity to look at these cells in real time. So I'll show you some of that dynamic data. Take this mouse, for example. We can monitor specific areas of the bone marrow with really high temporal resolution. Take this area here, and that allows us to visualize the real-time behavior of the bone marrow space. And hopefully that video is playing. What you can see here is the really stark static nature of these bone marrow clusters. So if I remind you earlier of the FACS data that I showed, showing how these brighter, larger static cells are our CD138 positive plasma cells. And this is in contrast to our far more dynamic smaller cells, which are our other post-germinal center B cells, including memory B cells. And below, you can see some of the quantification of that data, showing the real stark difference between the dynamic nature of these plasma cells versus other cells in the bone marrow. So what I really wanted to know was what was causing this unique clustering phenotype that we were observing. Was there a particular genetic profile at play? Was there something that we could target therapeutically, for example, to disrupt this niche and therefore increase the efficacy at which we could treat SLE? So in order to do that, we turn to single cell RNA-seq data. And in a collaboration with Shalin, Tony Pappenfuss and Ramya Malania, we generated some single cell RNA-seq data. And the big thing I want to understand here is how the signatures of these different cell types listed on that first dot point, for example, compare to one another. So, for example, what makes the autoimmune plasma cells different from the healthy? What are the differences between those smaller, fast-moving cells and those larger static cells? We wanted to investigate also if there's a difference between the plasma cells that expand and cluster versus those that don't. So in order to do that, just briefly on the technique, the skull has some real clear landmarks, which we use, such as the central sutures, to organize the area in which we image on imaging day. And then upon completion of the imaging, we're able to euthanize the mouse and really finally cut out that area, isolate the cells, and then perform single cell RNA-seq. And I've just got some very preliminary data to show you from that today. And essentially, when comparing the large autoimmune plasma cells to the smaller dynamic cells, for example, we had a list of about 100 differentially expressed genes that came back, and these included BLIMP1, XPB1, and a few others shown here, including various immunoglobulin genes, as well as a number of genes that are related to cell mobility and adhesion, including on the right here, actin beta, CCL5, Ly6A, Ly6C1, stuff like that. So it's possible here for us to try and identify some genes that we could potentially target therapeutically to disrupt this niche and make the plasma cells more susceptible to therapy. Just in my final few slides, I just want to say that when we look across the populations in these different mouse models, we notice a clear difference in how these cells expand in the disease versus healthy setting. So that's really highlighted here on the left. You can see a relatively random profile versus this more consistent profile on the right when we group particular confetti phenotypes. So for example, you can see a proportion of these RFP labeled cells are showing a consistent recombination pattern when we look at the VDJ genes. And that's the same with, for example, some of the CFP cells. So one final technique that I just want to talk about before I end my talk there is just to show how we can also look at the long bones. So in order to confirm a lot of these ligands that are highlighted by our sequencing data, we wanted to investigate with long bone analysis. And this is a technique that's really beautifully shown in a study published by McGuire and colleagues in Nature Comms. And essentially what this technique allows us to do is to take the long bones of these sick mice after imaging, shave off the sides so we're able to image through to the bone marrow space and get a really clear look at what's going on in these long bones. So this is a bone from a different study just to show you the type of transfer and the way it looks. And I've just got a 3D reconstruction here of one of our confetti positive bones. So you can see here that the data set's zooming in on some of these clusters. So here we have, for example, a YFP cluster. The beauty of this technique as well is it allows us to stain to different markers of interest. So here in the shown in purple, we have CD31 staining, which shows us the vasculature, so we can get information on how these cells interact with our vascular profile. Yeah, and again, we're able to move through and see this consistent phenotype where we see these single color dominated areas within the long bones in these sick mice. So that kind of just leads me to thank everybody that's been involved in the project, including everybody that was involved with sequencing, and also obviously mainly my supervisor, Edwin Hawkins, for his help with a lot of this work. And thank you for listening.
Excellent. Thanks so much, Joel. That's a really tour de force. So I'm just wondering whether we have any specific questions for your talk. As they come in, I guess I was really taken by the time course quantification, that aspect, and how you were able to, you were seeing the disruption of the niches. I guess you're really, where are you going with this? Like, what's the next step in your study? Yeah. Yeah.
So I tried to just keep it to the main points, but essentially, what we see, which I wasn't able to show today, is when we treat these mice therapeutically with these common treatment regimes that patients receive, such as glucocorticosteroid treatment, for example, dexamethasone, we see a real preferential resistance of these clusters to the therapy. So the idea that these clusters are disrupting in some way the effectiveness of the drug to treat and clear these autoimmune disease-driving cells. So the hypothesis is that if we can somehow identify what markers or what is exactly leading to this static kind of clustering phenotype, and we can somehow disrupt it, then existing therapies will be able to do a more adequate job of clearing these pathogenic populations, and we'll be able to get around these problems, such as in therapy, like when we treat patients, we just get kind of short-term symptomatic treatment and returning of disease, able to more comprehensively treat the disease population.
Yeah, cool. Well, good luck on that next step, as it would be.
Thank you.
sounds simple. Just fold it over in 1 year, right?
Yeah,
one afternoon. Okay, so I think we might bring everyone back to the table, as it would be. Shalin?
Yeah, thanks, Helen. That was incredible. Thanks so much to all of the speakers. I think we covered everything from perturb-seq to velocity to spatial to in vivo microscopy, ataxic, I mean, everything to get different flavors of the immune cell biology. And I think for me, that was the most fascinating aspect. So I'm gonna talk about clones, because that's an area that's close to my heart, and I think something that you all touched on, but I'd like to dig a little bit deeper there. So I think, at least for this particular set of talks, Joel's talk really highlights the size of the clones, the behavior of the clones, small, large, and then obviously those clones can change over time. Some might start small and stay small. Some might start small and get big. And we know that the same thing happens both in immune responses, which is relevant to Ash's work, but also presumably T-cell progenitors themselves will have heterogeneity in clone size. So when we take snapshots of these RNA-seq data, and it's great that we're getting these pseudo trajectories and populations we can culture, we are losing that clonal nuance, aren't we, in that some might be dead ends in terms of clones, but they'll contribute to our pseudo trajectory. So how are we going to separate those, and do we care? So I'll start with Ellen, and then Ash, and then Joel. So Ellen, please, yeah, give us your thoughts.
We agree with you 100%, Shalin, and actually we wanted very, very much to get the clonal data linked with the transcriptomes through some kind of a split clone approach, because this is such a lovely in vitro differentiation system that really should be possible. Unfortunately, with the COVID shutdowns and things like that, we didn't get our this lovely confocal microscope to do those experiments until fairly late in the day. But it's one of the reasons why we were very anxious to develop the seq-fish technology for exactly that purpose, so that we could look at individual little microclones in a well and score them according to gene expression profile. Because you're absolutely right. With our single cell reporters for BCL11B, which is the one gene we've really followed, it's a very important T cell transcription factor. We know that the timing of activation of that important factor can be heterogeneous right within a clone. So you plate single cells and you look at the progeny, and even though a clone in general can be either more advanced or less advanced, the individual cells in the clone activate the BCL11B at different times, even sometimes siblings. So I think that that's telling us that this could be hugely important for the choices they make. That's why I was so excited about Ashraful's data about the siblings. I think this is super interesting, and we wish we had a moral equivalent of TCR, unique clonal rearrangements.
Yeah, absolutely.
But being able to see the natural environment in Joel's work was just stunning. I thought these were gorgeous talks. So anyway, enough from
No, I think that's a great point that you raised, that you can't quite use the TCR, in the thymic study. So you're going to either have to use reporters or maybe lineage tracing barcodes or something like that. Yeah. So Ash, then I guess the natural question for you is, and maybe picking up on what Dawn asked you during at the end of your talk, you showed these arrows between the clonal siblings. Now, a single line can join two, but my first question is, did you have some clones that were more than two? And my second question is, did you categorize or try and lump them into a number of clones that had asymmetric decisions, symmetric in one, symmetric versus the other? That's part two of my question. And part three is, when was that decision of potency, let's call it for simplicity, made? Right at the beginning or a little bit downstream?
Three questions. You expect me to remember three questions?
Yes, I do. So part one, I can't remember.
Okay. Okay, let's hold on. So yes, they calculate the number of members of a family, and for the number of cells that we looked at, of course, we're only hundreds of cells, not tens of thousands, but we saw a range, most of them in families of 2 or 3, and then diminishing 4 and 5. We then had enough families to ask the question, to what extent do cells within the same family, if they're related, choose between one fate or the other? And what was interesting to us was that so we worked with mathematical modelers at UNSW, Miles Davenport and David Curry. They generated a random binomial model, and then found that the distribution of fates was not random, and that there's I would say, a modest predisposition for every family to move towards one fate and not the other. It's not absolute, but there's definitely a statistically significant predisposition to one fate or the other. And we couldn't explore that in the paper, but of course we want to, and we would hypothesize that it's really local interaction, where you are at that time, that dictates that fate. And we would love to see that, visually, using the techniques from Joel or Ellen, seq-fish, or watching individual clones in real time. And then your third question was, where is the choice made? And it is made, we believe, towards the end of clonal expansion. There is an overlap between decision to differentiate and the end of division. So I've always been a big fan of Phil Hodgkin's work. I don't necessarily always understand all of it, but I do like this idea that there's a decision to divide or a decision to differentiate. And in actual fact, at a population level, that those processes overlap. But we could see, for example, if we switched off TCF7, which is really required pretty early in the T cell activation, the cells very heavily go towards Th1 fate and could probably continue along that. So we would say that fate is, at least in our system, which is very reductionist, is early.
Yeah. Okay. Yeah. I mean, and this kind of points to some of the work that Alan Klein's been doing and Sam Morris and others, which is that these what they call hidden variables, which is these early fate decisions, which might be just because a transcript is not abundant does not make it important, and the RNA-seq is just pulling out what the most differences are. But if you can associate a clone's fate with gene expression early, then hopefully you can tease apart those other approaches. So it'll be fascinating to see, I think, in all of your systems, what those early decision controllers are. Which leads me to ask you, Joel, in your, did you track any of these clones? I think you did track these clones longitudinally. Did you see some clones, did you see heterogeneity in behavior over time? Some start small, stay small, some start small, go big, or vice versa?
Yeah, so I mean, the great thing about our technique is the ability to really, instead of getting that snapshot, is looking at them over time. So a common experiment I'll do is I'll monitor over about a week or two, kind of coming back every 2 days to see how these clusters develop, expand, and change. And the overwhelmingly consistent result is that these large kind of static CD138 positive cells are so consistent. Like we don't see significant expansion or reduction over time. Which suggests to us that this biology is from the development of these, a very long kind of a rare seeding event and very long-term slow expansion into these much larger clusters. And we see, we look at younger mice, but generally speaking, the clusters are much rarer or much smaller, and we look in the older mice and they get much larger and much more common. So yeah, just the idea that they aren't, the things do move slowly, and it does take a really long time to kind of develop these areas.
And just as a tack-on question to that, I guess it's difficult. I can't think of a way unless you do mass spec of the antibodies or something, but can you correlate clone size, clone behavior, RNA-seq with the amount of antibody produced in the serum of these mice?
I would love to. I don't really know how we could go about something like that. I'm sure better minds than I might have some better ideas. But yeah, the idea of like actually being able to show the correlation between like a given cell and its contribution to the autoantibody levels would be amazing, but I'm not quite sure exactly how we could go about it.
Yeah, and I guess there's a lot of bones in the body. You're not imaging all of them, just a couple in the calvarium, so it'd be a bit tough. Ellen, you were muted, but you had some enthusiastic response to something that was said. Did you have something to add?
I was basically agreeing very much. I think that these questions about lineage and microenvironment are incredibly important. And what really excites me about the whole single cell field and the ability to bring these technologies together is that I think that we're going to get some surprising answers from what controls them. So I'm very, very enthusiastic about this. I just agree with you about the lineage tracing importance, but also like with Joel's clones, I was just wondering if any of the same, if you could tell from, if you have any information yet, whether the same clone is both in the calvarium and in a long bone. I mean, I guess you PCR rearrangement data on that, but I'm just curious if you have that data at all yet.
Yeah, so up until this point, we don't, I mean, that's a very interesting question. That's exactly something we'd like to look at. Up until this point, most of the sequencing data has been done and isolated from calvarium samples. We haven't really done that comprehensive isolation from many different compartments from the same mice to look for consistent type of, VDJ arrangement, expression and stuff like that. But yeah, it's definitely something that I think would be great to look into.
Yeah, thanks.
Go for it, Ash.
Go, Ash.
I just wanted to know if you just did BCR sequencing for every single bone in the body of the mouse, would you find that you get radically different clones in each bone? Is that already known?
Yeah, so I'm not 100% sure, but I would say, like, from what we've seen is, I mean, there are a number of different clones and a number of different plasma cells in each bone marrow compartment, but it's always a few that seem to make up a bulk of the population. So we have quite a large variation in total, but when you break it down by contribution to that whole plasma cell population, we have a handful of a few clones that are so consistent in that representation. So the idea that it is very much dominated by the expansion of particular clones.
Right, so the next thing I wanted to ask is a technical question, and I really want to kind of applaud Ellen and Ash for using SmartSeq and deeper sequencing for these single cells to get at some of these low transcript factors, and the seq-fish, you know, let's call it a validation, or at least an orthogonal approach to looking at the gene counts for these lowly expressed genes. My question to you, Ellen, is did you try any of the kind of single cell RNA-seq inference methods like magic from Dana Pear? And I know there are a few of these which say, oh, well, let's just kind of use the averages and the relationships to kind of pull out those lowly expressed genes. And did you learn anything with SmartSeq that couldn't have been done with that?
Well, we actually did play with magic quite a lot, and it was very nice in giving us a lot of confidence about what the basic identities of the clusters were. So if what you're trying to do is to categorize the clusters, you don't need to have every transcript. But our agenda is really about causality. And for that, we need to know, does a cell that's expressing this gene need to have GATA3 turned on yet or not? So for that, we actually have to be able to measure whether they express GATA3. It doesn't do to just say it has a 80% likelihood that it probably expresses some GATA3. We have to actually be able to measure it. So we were very happy. Now, we used a fairly archaic version of seq-fish because Wen, before she came to my lab as a grad student, worked as a technician in Long Kai's lab. And many of you may know that seq-fish is sort of the West Coast version of merfish. They're very, very similar techniques. And now they've both been elaborated to whole genome scale, so you can do a lot with it. But I think that still we found that for transcription factor genes that you're trying to count in small cells, which don't have very much cytoplasm, so the pixels are all on top of each other, actually you don't want the high abundance genes there in the same probe mix, because otherwise you basically just run into a terrible overlay problem. So we were very happy with using seq-fish in this complementary way, letting 10x do the heavy lifting for the whole transcriptome and letting SmartSeq confirm it on smaller numbers of cells, but at the deep whole genome sequencing level. So I was happy that we had a chance to do all three.
Yeah, I think it was a fantastic addition. I think a lot of people really valued that kind of deeper investigation into these low express markers. And Ash, what about you? Did you investigate whether, you know, there's this debate going on. It's like, do you really need to capture all the mRNA of these cells, or can you just get more cells and infer it? Do you have any experience with that?
Yes. So we knew that dropout might be an issue for droplet-based work, and we played around with imputation not for the malaria work, but a different model, graft versus host disease, particularly in a preclinical model, where we wanted to look at the emergence relatively early of certain cytokines produced by those allogeneic T cells. And you know, even for the numbers of cells that we had, and it might have been as much as, I don't know, 50, 60,000 cells, the cells that we were most interested in were relatively rare. So yes, we did engage in using algorithms like MAGIC or ALRA, I think was the one that we used most frequently. And yes, it gave us an ability to extend our inferences, but it was very speculative, and I was quite reluctant, as someone who didn't really understand what was going on under the hood of those imputation algorithms. I didn't want to write a sentence in a paper that was based on that. So that's as far as we went. We didn't do any cross-referencing, but actually you raise a good point. We do have parallel SmartSeq 2 and chromium-based data sets for the malaria model, and it might be useful actually to do some cross-testing. So
I guess the other thing you could do is, because you've got this deep sequencing, you could computationally delete some of the UMIs for molecules to kind of mimic a 10x like data set and ask the question, did a full SmartSeq style data give you, could you use inference with the artificially deleted data set to infer it? Yeah.
You could do that. I guess our approach had been more sort of brute force, which was just sequence more cells. Yeah. And hopefully the stats will bear out that one of your molecules of interest will be higher expressed in one cluster than another cluster, and then you can go back into the wet lab and test that. So
and Joel, I know you've just started investigating the single-cell RNA-seq data for your clones, but do you have is anything jumping out at you in terms of intraclonal heterogeneity versus interclonal heterogeneity?
When it comes to the sequencing stuff, I'm very much more of a user than an expert, that's for sure. And we have kind of only been able to preliminarily kind of look at a lot of these results. But in general, when it comes to we are kind of more interested in the interclonal heterogeneity of the cells, kind of understanding why the cells as a group behave as they do rather than the intraclonal. I do think that, I mean, we have the data to look into, but I don't really have any answers for you off the bat about any particular things of interest.
Yeah, but it'll be really fascinating. Yeah, I think we're all really excited to see those results there. Ash, go ahead.
I just wanted to ask Joel, I believe that your system was sort of geared to looking at CD138 plasma cells.
Yeah, definitely.
Do you have is it possible to look at more short-lived plasmablast phenotypes in your model to sort of get at Shalin's question of heterogeneity within clones?
Yeah. Well, our model, to pair it with the imaging data, our model kind of relies on the post-germinal center expression, the AID of the germinal center to turn on so our plasmablast wouldn't kind of have that expression. I think, with the right model definitely could, and pair it with the technique and the data that we have to answer those questions, but not in the current data sets that we have, I don't think so.
I'm going to sneak in one last technical question for Ash, which is this spatial RNA-seq data. And I think this is going to be a challenge for a lot of people, is to say, well, until we get to single-cell spatial, I think someone had a question actually, which I'll paraphrase in my question, which is, firstly, this is going to be a huge challenge, right? To separate beads that are related to one or multiple cells. And can you use what Ido Amit used for PIC-seq, which is this paired immune cell data? Because there he gets single cell RNA-seq data on the population, then he sorts the doublets and gets the paired immune cell data. So in some sense, if you, in a separate mouse or a different part of the spleen, got your single cell RNA-seq data, then had your spatial data, could you computationally calculate which are doublets, triplets, etc., and tease it apart that way?
Absolutely. So what we did for this, or doing rather for this system, for this project, is to generate a parallel droplet-based single-cell RNA sequencing data set, which we can then use to perform exactly what you've just suggested. The problem with that is that we only have some cells. So it's all immune cell data. We haven't gone back and harvested and generated a stromal cell data set, but we have collaborators who have got that. So what we're doing is integrating across droplet-based data sets, generating a single atlas, and then importing that and using that within the spatial data. So we'll keep you posted.
Yeah, look, I think the world is, I think our world is really thinking about this a lot.
Yeah, the world is probably thinking about a few other things right now.
A few other things, exactly. Look, on that note, Ellen, Ashraful, Joel, it was a fantastic session. I had so much fun, and I wish we could talk longer. Unfortunately, our time has come to an end. So just to thank you for your time and your wonderful presentations. and we wish you the best. So
Your wonderful questions, which raised the level of all of us. I mean, I think you sent us home with lots to think about.
Good. You've got your homework.
That's it. So look, I think we can all sign off now, and please stay online. Helen's going to talk us to the audience through on what the next episodes are, including the Oz single cell poster session, for which there are prizes. So long, our panelists. Thank you very much again.
Great.
Bye.
All right, so I will just very, very briefly talk us through what's remaining coming up in our next sessions. So before I do that, actually, I would like to just remind everyone that you can catch up on the previous webinars at this web address. So this is actually our 9th episode, and we do thank our very many wonderful series sponsors for making this all happen. And so the next episode that's planned in the series is a computational biology discussion about deeper insights into single cells. And so mark it in your diary. It's on the 18th of November for the 11 a.m. Sydney equivalent time and Melbourne equivalent time. And you can register at that tiny URL. We'll also send out emails to the registrants of today, and you'll see some advertising on Twitter, as we usually like to do. So with that, as Shalin had mentioned, we're also showcasing your research. And the way we're going to do that this year, in light of not being able to run a traditional traditional conference, is to actually run a virtual poster session. And so we're encouraging you to submit your posters. We'll be engaging, it'll be an opportunity for you to engage with researchers, and you'll also be the option to submit a poster as well as a video to describe your research. And don't forget that we will be awarding cash prizes for both the best poster and video. So you can see the registration here. Again, keep an eye on our Twitter feed, as well as some mail outs that will be sent, and a deadline, of course, of the 30th of November. So thanks everyone for joining, and we'll see you next episode.